Hi, I'm David and this is the Biology Classroom. This is a paper discussion video, so get ready with this paper and let's go through the questions and answers together. Question 1 is about an organism called the Bactrian camel. A. Suggests reasons why the wild Bactrian camel has become critically endangered. You can use the common reasons why an organism becomes endangered to answer this question, but make sure your points are related to this example. First, habitat loss due to human activities. For example, land used to build roads. 2. Competition with farm animals or alien species. Alien species are organisms that entered and established in the environment outside their natural habitat. 3. Climate change. 4. Hunting, poaching, or killing by humans. There is 1 mark for AVP. Any logical examples with proper elaboration can give you this mark. For example, their breeding sites are used for illegal mining. B wants you to outline the role of the IUCN. They are a global, international, or worldwide authority or advisory. They give advice to conserve and protect nature, biodiversity, or habitats. They also assess and categorize species conservation status. IUCN creates the Red List of threatened species. This information can influence governments on the policy related to conservation. The information they have can also be used to educate or raise awareness of the public on endangered species. C talks about assisted reproduction techniques zoos use for breeding programs for endangered species. You are asked to describe the embryo transfer procedure in a mammal such as a camel. The first point is any method to obtain embryos. The animals may be allowed to mate naturally, or techniques such as IVF and artificial insemination can be done. Then, remove the embryo from the mated or artificially inseminated female. Selection is done to choose the healthy, normal, or best embryos. Some of them may be frozen for storage or later use. After that, implant embryos in the uterus of a different female. These females are called surrogate mothers. Surrogate mothers receive hormonal treatment prior to the procedure to prepare for pregnancy. Question 2 is about photosynthesis. A. Name 1 chloroplast pigment. One of these names can give you a mark, but be specific. Instead of only saying chlorophyll, write chlorophyll A or B. B. Explain the relationship between the absorption spectrum of the main chloroplast pigments in a species of plant and the action spectrum. You need to know what the graphs show you to answer this question. The action spectrum shows the rate of photosynthesis of a plant at different light wavelengths or color. The absorption spectrum shows how much each wavelength or color is absorbed by the pigments. If the pigments in a plant absorb more wavelength, more energy is available for the light-independent stage of photosynthesis. So, higher absorption gives higher photosynthesis. C. Explain why temperature can be a limiting factor of photosynthesis. First of all, remember that photosynthesis is an enzymatic reaction. You can use your AS knowledge of how the rate of enzymatic reactions is affected by temperature to answer this question. Higher temperature increases the rate of enzymatic reactions by affecting the rate of enzyme-substrate complexes formation. You can also use the specific stages or events in photosynthesis to get this point. For example, the photophosphorylation, light-dependent stage, or light-independent stage. These enzymes denatured above optimum temperature. 1 AVP is given here. For example, if you mention that rubisco is an enzyme involved in the Calvin cycle. D wants you to outline how the light-independent stage of photosynthesis leads to the production of carbohydrates such as starch in plant leaves. The first step is carbon fixation, where RuBP joins with carbon dioxide to make a 6-carbon intermediate. It will then split into 2 molecules of GP. GP will then be reduced to TP or triose phosphate. This step uses ATP and reduces NADP. Some carbons in TP are used to make glucose. When many glucose is synthesized, condensation occurs, where glycosidic bonds form between glucose to make starch. Question 3 is about an investigation involving mitochondria. They remove the outer membrane of the mitochondria and then analyze the inner membrane and the matrix of mitochondria. In question 1, the outer and inner membrane of mitochondria was placed in pure water. The outer membrane ruptured, but the inner membrane did not. You are asked to suggest and explain why the inner membrane of a mitochondrion remains intact when the organelle is placed in pure water. Since the inner membrane folds into many cristae, when water enters by osmosis, it will expand but not burst. Another possibility is that the inner membrane is not as permeable to water, so not much water moves in by osmosis. Lastly, the inner membrane may move ions out of the matrix. This causes the matrix to have a higher water potential, so less water enters by osmosis. Question 2 wants you to name 3 molecules other than the core enzymes found in the mitochondrial matrix. You also have to explain their role in aerobic respiration. Number 1, pyruvate. The product of glycolysis will be found here. It is for the link reaction to occur. You can also talk about the purpose of link reaction to get this point. Those substances involved in the Krebs cycle will be found here as well. This includes oxaloacetate, to accept acetal or make citrate. Next is citrate, to make reduced NAD or to be dehydrogenated. You can also find all the enzymes required to catalyze link reaction or Krebs cycle. Oxygen is found here as they are the final electron acceptor in ETC. Water must be present as well. It is a solvent or medium for reactions to take place. Lastly, circular DNA and the RNA synthesized from them. This is to make respiratory enzymes and other proteins needed in mitochondria. 3 says that a high proportion of the molecule cardiolipin is in the inner membrane. It makes the membrane impermeable to some ions. You need to suggest the reason for this. The inner membrane contains many proton pumps. Cardiolipin helps prevent the protons pumped out by these proteins from returning to the matrix. Hence, a proton gradient can be maintained for chemiosmosis to occur. In B, you have another experiment. It was found that the matrix has a negative charge. This membrane potential is also the driving force of the transport of ATP, ADP, and inorganic phosphate. Question 1 wants you to explain how hydrogen atoms from reduced NAD and reduced FAD lead to a membrane potential forming across the inner mitochondrial membrane during oxidative phosphorylation. This question can be answered by using the steps in the oxidative phosphorylation. The hydrogen atoms released by reduced NAD and FAD split into protons and electrons. Electrons flow or move down ETC. They release energy, which is used by the proteins to move proton to the intermembrane space. There will be a buildup of proton in the intermembrane space. It sets up a proton or electrochemical gradient. Question 2. Suggest and explain how inorganic phosphate is transported across the inner membrane of the mitochondrion into the matrix. You can see from the diagram that it is transported by facilitated diffusion or through a protein channel or carrier. Inorganic phosphate is charged, so a transport protein is always needed. The diagram also shows co-transportation as inorganic phosphate and proton move together. When we learn the process of chemiosmosis, we say that protons enter the matrix via the ATP synthase. So, this is the third point. Question 3 wants you to suggest the advantages of linking ATP transport to ADP transport across the inner membrane of mitochondria. In the diagram, you can see that every time ATP is made and transported out of the matrix, ADP is being transported in. This ensures a constant supply of ADP or reactant to enter the matrix, so ATP can continue to be made. Question 4 is about ADA deficiency, an immune system disorder caused by a recessive autosomal mutation. It can cause a condition known as ADA-SCID. A wants you to outline the principles of genetic engineering. Even though this is a very complicated and tedious procedure, the question says outline, and there are only 4 marks. You should only give the main stages involved. Detailed elaboration is not required. First, this procedure aims to add DNA to a cell or organism to give a new characteristic or protein. Then, briefly describe the role of one of the most essential enzymes in genetic engineering, the restriction enzyme. No mark is given if you only mention the name. After that, insert the gene into the vector or plasmid. The recombinant vector is then introduced into the host cell, such as bacterium. After the transformation, clone or multiply the GM organism. There is one mark for AVP. For example, you can provide the role of ligase in making recombinant DNA or the method used to transform the host cell. B shows you the steps involved in gene therapy. Question 1. Explain why a single infusion of gene-corrected stem cells is enough to cure the disease. A stem cell can proliferate or divide indefinitely to make new cells. So, we don't have to repeatedly inject the gene-corrected stem cells into the patient, as the new cells they form would inherit the gene inserted into them. 2. Explain why a promoter has to be transferred as well as the desired gene. This is a very commonly asked question in paper 4. First, it switches the gene on, so the gene is expressed. In other words, it allows transcription of the gene to occur. Promoter can also regulate the timing and the rate of gene expression. We can use it to allow the expression only at a certain time or all the time and in sufficient quantities. Lastly, a promoter is the binding site of RNA polymerase and transcription factors. 3. State 2 ethical considerations of using a retrovirus for gene therapy. Viral vectors can be problematic when used in gene therapy. Some risks must be assessed before they are used. Firstly, the new gene could be inserted by them into the wrong place, causing disruption to other genes. Some viral vectors could cause cancer in the patient. We also need to ensure the selected viral vector does not cause infection or disease in the receiver. Lastly, there is a risk of immune response or allergic response. Question C wants you to explain why gene editing is more suitable as a potential cure for Huntington's disease. Gene therapy inserts a functional gene into a cell or an organism, so it wouldn't work on a disease caused by a dominant allele. Adding the normal recessive allele could not eliminate the disease-causing dominant allele. Gene editing, on the other hand, can delete, remove, or inactivate the dominant allele that causes Huntington's disease. The surplus CAG repeats, called stutter, can be deleted using this procedure, making the allele normal and non-disease causing again. Question 5 gives you some information about alpine plant species in New Zealand. A tells us that the alpine areas nearest the South Island are 1,500 to 2,000 km away across the sea. These species are not found anywhere else in the world. So, how can these species develop on the South Island? It is probably due to evolution or speciation as a part of natural selection. Random or spontaneous mutations occurred in some of the plants that existed on the South Island. This caused some plants to adapt to the alpine habitat, which means high altitude or mountains. They survived and were selected for by the environment. The selection pressure in these habitats is the cold, strong winds, and thin soil. They must survive and reproduce, and those alleles for alpine features are passed on to their offspring. This leads to a change in the population's allele frequency or gene pool. Eventually, the population becomes morphologically, physiologically, or biochemically different, forming a new species. With these adaptations, they have a higher chance of survival since there are fewer herbivores and competition in this new niche they now occupy. B shows you a graph of mountain's mean height and the tree line's height over time. In the graph, you can see that the mean height of mountains keep increasing over time. At one point, it exceeds the tree line. 1. Identify with reasons the time period when South Island's alpine plant species developed. The alpine species developed when an alpine habitat was formed. The graph shows that it is around 0.95 million years from now. This is when mountains are always higher than the tree line, creating this unique alpine habitat where regular trees won't survive. 2. Suggests how DNA sequence data could be used to confirm the time period you identify in 1. We can use the technique to find the similarities and differences in DNA between alpine species and related lowland species. Then, using the molecular clock, we can estimate the time when species evolved or separated and diverged from each other, meaning that speciation has occurred. If there are many based on nucleotide differences, many mutations have occurred. This means they separated long ago and have a more distant common ancestor. 6a wants you to outline how vigorous, uniform varieties of maize were developed by selective breeding. Your answer to this question should be brief but organized logically to show how the procedure is carried out. Firstly, breed or cross plants with desired characteristic. You can get this mark if you use a specific example of the characteristic. For instance, one group can produce more kernels and another group that has a higher nutritional value. When the offspring are produced, breed the best offspring repeatedly for many generations for both groups. This is known as inbreeding or line breeding. The two inbred lines are then hybridized. You can also use the word outbreed or provide a description. The hybridization of the two lines gives hybrid vigor, heterosis, or outbreeding enhancement in the resulting offspring. The F1 hybrid will have the same genotype, hence a uniform phenotype. In B, you have the information on IR8, which is a product of the breeding between another two variants. You can see from the diagram that IR8 has a height in between the two parental variants. Question 1. Suggest why IR8 is an improvement on the Peta variety of rice. Since it is shorter, it is less likely to fall over or collapse. This usually happens due to the weather conditions when a plant is too tall. Secondly, since the plant uses less energy to grow taller, it may produce more edible grain. Question 2 is about another variety, IR64 sub1. It is by the breeding between FR13A and IR64. The question wants you to suggest the benefit of sequencing the DNA of IR64 sub1. Sequencing DNA means determining the order of nucleotides or bases that make up the DNA molecule. By sequencing the DNA of this species, they can check for genes for height yield and flood tolerance this hybrid inherited from the parental varieties. Question 3. Explain why the scientists cross IR64 sub1 with IR64. Since they both have high yields, crossing them may maintain or even increase their yield further. Another reason is that we occasionally outbreed a group to prevent inbreeding depression. C shows you a plant that has dwarf varieties and an experiment where they are treated with gibberellin. Both wild type and variety A become taller after the treatment, but B shows no response. Question 1. Suggest explanations for the different responses to gibberellin shown by dwarf varieties A and B. To answer this question, you need to describe the action of gibberellin that causes a plant to increase in height. In A, gibberellins bind to a receptor called GID1 on the cell surface membrane of the cells. This causes the DELA protein to be broken down. PIF or transcription factors are now released to bind to DNA, starting the transcription of the gene. The expression of those genes leads to stem or internode or cell elongation. The genotype of A is homozygous recessive. It has no dominant allele. It does not produce its own active gibberellin. Hence, when gibberellin is applied, it can grow taller. B does not respond to the plant hormones. It probably does not have a functioning gibberellin receptor. So, the application of gibberellin does not cause any changes. Question 2 wants you to outline the features of a transcription factor such as BZR1. A transcription factor binds to the promoter and allows gene expression. It allows the binding of RNA polymerase to the promoter, which initiates the transcription process. Question 7a is about a genetic diagram. The gene controlling the fur color of cats is located on the X chromosome. There are two alleles, black and ginger. The two alleles are codominant. A heterozygous cat would be a tortoiseshell. You need to show the results of a cross between a female tortoiseshell cat and a male ginger cat. Remember to use the letter X to represent the X linkage when you choose the symbol. Then, use two other letters to show the allele. A tortoiseshell female would be heterozygous. Male cats have an X and Y chromosome. This individual is ginger, so it is XG and Y. For gametes, separate all the alleles and circle each of them. Combine the alleles to get the 4 possibilities. Then, identify both the color of the fur and the gender of the individual. B wants you to describe how the expression of the TYR gene leads to the production of melanin. TYR gene codes for tyrosinase. This enzyme catalyzes the conversion of tyrosine into DOPA. Then, DOPA is converted into dopaquinone. Dopaquinone is used to make melanin. In A, you have to name the structure or region labeled A to C in the diagram. A is the segment before the collecting duct, the distal convoluted tubule. Please write the full name, not just DCT. B is the region nearer to the surface of the kidney. It is the cortex. C is the U-shaped segment between PCT and DCT, the lobe of Henle. B shows the relationship between blood ADH concentration, urine concentration, and the flow rate of urine. You are asked to describe the relationship shown in figure 8.2. Firstly, give the general statements describing how ADH concentrations affect the two variables on the y-axis. One of them increases and the other decreases as ADH concentration increases. The two lines also show that urine concentration increases as the urine flow rate decreases. Then, quote in data with units. For example, when ADH concentration increases from 10 to 90 arbitrary units, the urine concentration increases by 990 millimole per dm cube. I usually prefer to do a bit of calculation rather than quote the data directly because some questions expect you to show manipulated data, not just straightforward quoting. C. Describe and explain the action of ADH on the cells of the collecting duct when the water potential of the blood decreases. When you talk about the action of ADH, make sure you use the names of all the structure and substrate involved and sequentially arrange your points. When ADH arrives at the kidney, it binds to the receptor on the cell surface membrane of collecting duct cells. Then, G proteins and adenylate cyclase are activated. A second messenger called CAMP is produced. This leads to the activation of protein kinase. Vesicles that contain aquaporins move towards and fuse with the cell surface membrane. There will be more aquaporins on the cell surface membrane. Collecting duct cells become more permeable to water. Hence, more water leaves the lumen and is reabsorbed into the blood. In question 9a, we have a graph of action potential in a motor neuron and a graph of action potential in a striated muscle fiber. With reference to figure 9.1 and 9.2, describe the differences between the action potential in a motor neuron and the action potential in a striated muscle fiber. The first thing we can compare is the resting potential. It is higher in motor neuron. Quote in the values to get another mark. Then, compare the difference of potential when depolarization occurs. It is smaller for the motor neuron. Give the values here as well. The time taken for depolarization is shorter for motor neuron. Lastly, hyperpolarization or refractory period only occurs in the motor neuron. B has a graph of the tension in a striated muscle during the 3 contraction phases. The question explains that a muscle's tension represents its fiber's degree of contraction. The graph shows that the tension increases rapidly in the contraction phase and declines gradually in the relaxation phase. 1. Explain what is happening in the striated muscle fiber during the latent phase. This phase is right after the stimulation, before the muscle fiber contracts. To prepare muscle for contraction, the transverse or T-tubules must be depolarized. Calcium ion will then diffuse out of the sacroplasmic reticulum. They bind to troponin, causing it to change shape. Then, tropomyosin moves and exposes binding site on actin. These binding sites are for the myosin head to attach to actin. Cross bridges will form where the myosin head binds to actin. 2. Suggests why the relaxation phase shows a gradual decrease in muscle tension. Cross bridges may break gradually. All the myosin heads detach at different times. So, you won't have the muscle to relax suddenly and rapidly after the contraction. In question 10, we have a passage about homeostasis. Homeostasis is the mechanism to keep the body's internal environment in optimum conditions. The second paragraph discusses the decrease in corrective action when a condition has returned to its set point. This is the negative feedback mechanism. When glucose binds to the receptor on the pancreatic cells, the endocrine cells, called alpha and beta cells, will secrete insulin and glucagon respectively. Glucagon can stimulate the breakdown of glycogen into glucose. That's all for today. If you think my videos are useful, don't forget to like, comment and subscribe. Thank you for watching and see you again soon.